Good afternoon, student. Today I am going to start chapter 6, the structure of DNA. The structure of DNA. DNA stands for deoxyribose nucleic acid. Deoxyribonucleic acid. DNA present in nucleus of cell. Structure of a cell. Cell wall. In center there is a dense mass structures present. This is known as nucleus. And this portion is cytoplasm. Inside nucleus, nucleic acid is present. Nucleic acid is present. Inside nucleic acid there is thread-like structures present which form a condensed structures. Nucleic acid are of generally two types. One is also known as the ribose nucleic acid. And deoxyribose nucleic acid. Ribose nucleic acid is known as RNA and deoxyribose nucleic acid is DNA. Here we discuss the structure of DNA. DNA during cell division these chromatids condensed and form a thread-like structure. This thread-like structure is known as chromosome. These are the structures of chromosome. Single structure of chromosome. Inside chromosome there is double helical structures are also present. This structure is known as DNA gene. DNA present inside chromosome throughout their length. DNA carries genetic material which is transferred from generation to generation. Participate in the inheritance of characters. Now discuss DNA is a long chain of polynucleotides. Polynucleotides, long chain of polymer of polynucleotide. Polymer is a number of monomers from the polynucleotide chain. Of nucleotide. Nucleotide is composed of three compounds. Compound or components. In center, oxygen is present, and this is the pentose sugar. Pentose sugar, phosphate group, and here is base. The structure of a nucleotide. The structure of a nucleotide. Pentose sugar. Pentose sugar are of two types: ribose sugar and deoxyribose sugar. The structure of ribose sugar. One, two, three, and 4 V and 5 is outside the chain. Hydroxyl group present on 2 or 3 carbon chain, and this is known as ribose sugar. Hydroxyl group present on 3 carbon positions, this is known as deoxy sugar. So this is the structure of ribose sugar or deoxyribose sugar. A nitrogenous base. It is a nitrogen containing organic nucleus having similar physical properties of a base. Nitrogenous base are two types. One pyrimidine and second purine. Pyrimidine is cytosine, thymine, uracil. Purines, adenine, guanine. Cytosine common in both RNA and DNA, but uracil is present in RNA. When a nitrogenous base is linked with pentose sugar through N-glycosidic linkage to form a nucleoside. So many examples of nucleoside. Adenine plus ribose sugar, adenosine. Guanine plus ribose sugar, guanosine. Adenine plus deoxyribose sugar, deoxyadenosine. Cytosine plus ribose sugar, cytidine. Cytosine plus deoxyribose sugar, deoxycytidine. Thymine plus deoxyribose sugar, deoxythymidine. Uracil plus ribose sugar, uridine. Here is the structure of nucleoside. Pentose sugar attached with the base like this. Pentose sugar attached with base. Adenine, A stand for adenine, C stand for cytosine, G stand for guanine, T stand for thymine, and U stand for uracil. So this pentose sugar, linked with N-glycosidic bond to bases to form nucleoside. Phosphatase group. Phosphate group. A nucleotide is formed when a phosphate group linked to 5' OH of a nucleoside through a phosphoester linkage. Two nucleotides, when linked through a 3' to 5' direction phosphodiester linkage, form a dinucleotide. In a similar fashion, more nucleotides may join to form polynucleotide chain. The polynucleotide chain is formed. One end with free phosphate moiety of ribose sugar is called as 5' end of polynucleotide chain. The other end with free hydroxyl group marked as 3' end of polynucleotide chain. 5' end free phosphate and 3' end free hydroxyl molecule to form a 3' end and 5' end. Thus sugar and phosphate from the backbone in a polymer chain and the nitrogenous base linked to sugar moiety project from this backbone structure. Nitrogenous base pair complementary to each other with a hydrogen bond present in double and triple manner by a rule is known as Kaffer's rule. According to this rule, the amount of cytosine is always equal to guanine and the amount of adenine is always equal to thymine. That is, A is equal to T, C is equal to G. C joined with G with triple bond, A and A joined with T with double bond. Like A joined with T by double bond and C joined with G with the triple bond. The ratio of A stand for adenine and G stand for guanine. The ratio of adenine and guanine to that of cytosine and thymine is always equal to 1, like A plus G by T plus C is equal to 1. A structure of double strand polynucleotide chain. Polynucleotide chain. Phosphate and sugar both link to form double stranded polynucleotide chain structure. Base pair, hydrogen bond, hydroxyl end, 5' end, 3' end, 5' end, 3' end. The stand of polynucleotide chain is run anti-parallel to each other. Anti-parallel. Each other. 3' end, 5' end, 5' end, 3' end. The backbone, that is a stand, is made up of pentose sugar, base and phosphate. Base pair bind the two stand with each other with a hydrogen bond. So this parallel line T A. Two stand bind with each other and form the base pair like this. This is C and this is G. Same way, five dash and 3 dash and 5 dash and 3 dash and this is the pattern by which the two stand joint with each other with the help of nitrogenous base. The two strand is run parallel and joined with each other with a polarity and distance is equal to 20 angstrom. Now helical structure of DNA. The distance of one-third helix is 3.4 nm. Polarity of two strand is 20 angstrom and distance of two base pair zero point 0.34 nm. Adenine, thymine, guanine, cytosine. Polypeptide helix. Strands. Helical structure of